Welcome everyone to our ActorSting webinar for today. My name is Andrew Forbes from Monash University and I'm co-chair of ActorSting with Kate Lee from MCRI, who unfortunately couldn't make it today. We're delighted today to have our talk about data integrity and a tool for assessing data integrity, which is such an important topic in today's world of data, especially with AI and ability to manufacture things that are not real. First, I would like to begin by acknowledging the traditional custodians of the lands which we're all meeting. For me, it's the Bunurong people of the Kulin Nation, and pay respect to elders past and present, and also to those elders past and present on lands which all of you are currently located. Just a couple of housekeeping items here. This webinar is being recorded and it's going to be available to view on the Actor website, and there is an Actor YouTube channel, and so this will be featured on there. All of you are going to be placed on mute and all your cameras turned off by the higher power, Natalie Shalit, who's our Actor project manager in charge of ActorSting functions. And also the chat functions has been turned off. But the Q&A button is alive and well. So if you have questions during the talk, please put them in the Q&A and I'll read them out. Well, if it seems appropriate, I'll actually stop the presenters for a point of clarification, or I'll read out the question at the end, and we'll also have the usual question, the Q&A at the end of the talk as well. So please do use the Q&A either during the talk or at the end. So let's move on. I will introduce our presenters today, Kylie Hunter and Sol Liebsman. I hope I got the pronunciation right, yeah. Sol?
Sounds good.
Both from the Next Gen Evidence Synthesis team at the NHMRC Clinical Trial Centre. So Sol specialises in advancing the methods and models used in individual participant data meta-analysis and network meta-analysis. And Kylie Hunter is a research fellow and co-lead of this Next Gen Evidence Synthesis team. And her research centres on advancing evidence synthesis methods to address high priority research questions with a particular focus on individual participant data type analyses. So we're very interested to hear about a tool for data integrity. So I'll hand over to whoever's starting off from, both of you, Kylie or Sol. So thanks very much.
Thanks. I'll just confirm you can hear me okay?
Yes, that's great.
Excellent. Yeah, thanks so much for having us. We're very excited to share with you our tool for assessing integrity using individual participant data. So as an overview, I'm going to briefly talk about what the integrity crisis is in medical research, what has been done to date to address this. Then we're going to introduce our individual participant data integrity tool, and then talk about, well, what's next, and there'll be opportunity for questions and discussion. And we wanted to make this a little bit interactive, so hopefully this works. But if you have a phone, you can scan this QR code here. Otherwise, you can go on your computer to slido.com and enter in this code. And there will be a few questions throughout the presentation to make it interactive. So I'll just give people a moment. I can't see anyone in videos, so I don't know if you need more time, but maybe I will move on unless anyone says they need more time. I guess just to start, we wanted to gauge, who you are. So what areas do you have expertise in? Where we're guessing there's a few statisticians, clinical trialists, but if you could type as many that apply, just so we get some sort of an idea. Yeah, so statistics. Do we have any clinicians or methodologists as well? Economics, okay, great. Clinical trials, epidemiology. Okay. Astrophysics. Oh. This is good. We've got a nice broad verbosity. Preclinical data management. All right. No, that's great. Thank you for that. That's great to see who we've got here. In the interest of time, project management, I will move on. So there is an integrity crisis in medical research. Some people might think that's melodramatic, but I'll go through these slides and then you can decide for yourself. So in 2023 alone, more than 10,000 publications were retracted due to integrity issues. And we've seen a proliferation of articles on cases of fraud, research misconduct, research integrity issues, some of which are shown here. They just keep coming every day. There's new ones. So estimates vary, but experts say that, around 20% of research may be affected by research misconduct. Again, it's hard to know, and there's lots of different estimates, but if you actually, I guess it's not just media hype. If you ask researchers themselves about misconduct, there's some interesting findings. So this meta-analysis of 21 survey studies found that around 2% of scientists actually admitted to having fabricated or falsified data at least once, and 34% admitted to engaging in questionable research practices. So things like data dredging or inappropriate analyses. And then when they're asked about their colleagues, they said these estimates increased to 14% and 72% respectively. So what are the consequences of this? Well, if we have this untrustworthy research, we can consider this to be a pollutant which flows into our river of evidence and contaminates it. And then this flows on to inform our guidelines and policy and treatment decisions. And these consequences were illustrated in the case of ivermectin for the treatment of COVID. So millions of COVID patients around the world were administered this ivermectin drug based on a review of studies showing it could increase survival by 51%. So, pretty significant. There was a bit of excitement and hype. But it was later discovered that several of the studies underpinning this evidence were actually fraudulent or had major integrity issues. And when these studies were excluded from the analysis, there was no longer any effect on survival in these patients. So what we have here is patients that are being administered a drug without any benefit, but with known side effects. This led to shortages of ivermectin for other approved uses, also public mistrust in science, and a waste of resources that could have been redirected elsewhere. So in the words of James Heathers, this is an existential threat to the scientific enterprise. Briefly, another example of consequences, which we're probably quite familiar with, is the case of autism and vaccination. So in 1998, there was a study published in The Lancet, which linked the MMR vaccine to autism. Shortly thereafter, there was a substantial decline in vaccination rates, and this led to outbreaks. But in 2010, the study was retracted due to misconduct, but the damage is already done. This information had already been festering for 12 plus years, and today we know that vaccine hesitancy does really persist as a consequence. So I guess before we plow on, what is research integrity? It can be defined as adherence to the highest standards in conducting research or best practice. So that's shown on the right here in green. And on the other end of the spectrum, we have worst practice. So that's deliberate misconduct, and that comprises, fabrication, which is completely making up data, falsification, which is manipulating data or research materials, and plagiarism. And then again, in the middle here, we have the questionable research practices, which, as we saw from the survey result I showed earlier, is really prevalent and includes all these things listed here, such as inappropriate analyses. So just going to throw it back to you. Checking, have you ever had concerns about the integrity of a trial? So we've got never, sometimes, often, always. Hopefully no one selects always, but sometimes. Okay. All right. So we've got most people here saying sometimes, which I think is about right, but quite a few saying never as well. Okay, 1 in 5 saying never. It's good to see that no one says always, although maybe we do need some sort of skepticism. Okay, so I'll move on to what has been done to date to address this integrity crisis. So there has been many promising tools and developments. So these ones are used to assess the integrity of studies once they've been published. So using summary information, and they include the tracked checklist, there's the reappraise tool, Cochrane have a couple of things. So there are quite a few things emerging. But as I said, again, they look at summary data or publications. They're looking at things like retraction notices, trial registration, ethics approval, plausibility, bibliometrics. So I've got this image here. There's a Spanish chemist who in 2023 was publishing one study every 37 hours. So he was quite excessively prolific, and that is an integrity issue because either he's working nonstop or, yeah, it's made up. So, and I guess doing these integrity checks, it takes time. And so there are also emerging tools to make this processes more automatic, but I won't go into too much detail here. So I guess the limitation here is that all the tools I've shown you focus on publications and summary data, but a lot of integrity issues can only be detected by looking at raw data. So the individual participant data, the raw line by line data for each participant within a study. Even something as simple as copying and pasting rows of data, which does happen, you wouldn't know about it with summary data. You need the IPD, and it allows much more in-depth checks, which we'll go into a bit more soon. So to illustrate the power of IPD, editor John Carlisle, he examined trials submitted to his anesthesia journal and found that when raw data were not available, almost all studies appeared to be trustworthy, as shown here in blue. But when data became available to him, he detected a massive 44% of studies that included flawed data. So this is not what we want to be, basing our treatment decisions on. So a few years ago, our team at University of Sydney, we were conducting an individual participant data meta-analysis on cord management at preterm birth. And we knew, there was all these concerns about research integrity. So we thought, well, we need to assess trials. So my supervisor asked me, hey, Kylie, can you look into how we can do this? And I thought it would just be straightforward. There'd be some sort of a tool and off we go. But of course, there wasn't anything available at the time. No standardized tools to assess integrity of trials using IPD. So from that, I then began a large project as part of my PhD, which aimed to develop this IPD integrity tool to screen randomized trials for integrity issues. So now I think we're just going to check in on your previous experience. So have you ever conducted trial integrity checks in your role? And if you have, is it just using publications? Have you looked at IPD? Have you done both? So you can see here, everyone so far is a no. Okay, but we've got some using both. So maybe people's role doesn't require it, or maybe there's some people that might need to start doing some integrity checks. Let's see. So if we go to our next slide, oh yeah, so if you did use a systematic tool, did you use a systematic tool such as tracked or ad hoc checks? And we've got everyone so far saying ad hoc checks. Okay. Very interesting. All right. Well, thanks for.
Hi, Kylie. Sorry, it's Andrew here. Just can I just can you define what sort of data integrity checks you would think people might respond to here? What sort of checking methods might be used?
So I guess Sol's going to go into a few more of these shortly, but things like, the unusual data patterns, like you checking for terminal digit bias, or like logic checks, consistency checks, plausibility, things like that. So there is a bit of an overlap with general data checking and cleaning, with maybe checking if expected correlations are present, ranges, that type of thing.
Yeah, so some of them are routine, sort of for the clinical trial statistician you do anyway. Yeah,
but also then the publication level things as well. So are you looking for attraction notices or questions of concern, that type of thing? Yeah.
Okay. Yeah, thanks.
Okay, so I think now I'm going to hand over to Sol to talk a bit more about our tool. Over to you, Sol.
Thank you, Kylie. Hi guys, I'm Sol. So I guess interestingly, a lot of people are doing data checks of some variety. But I guess the problem Kylie encountered and then that we all engaged with as a team is a lot of them are ad hoc and there's a lot of heterogeneity there. And we really wanted to dive in and make a systematic tool that I guess covered what we throughout the project then uncovered to be the best items and checks to reveal untrustworthy data. And so I guess I'll start with how we developed the tool. We first started with a big literature review and we collated all the potential relevant data items that could potentially be included. And then from here, we took this huge myriad of items out there and we brought it to an expert advisory group and we had a discussion and developed the items that we thought would be most appropriate and that were deemed most relevant for our integrity tool. And then from here, once we had our prime candidate items, we kind of had our first generation of our integrity tool. And we pilot tested it and performed preliminary validation. And so we were running 2 IPD meta-analyses at the time, and they were 2 of the vessels to perform pilot testing. And we also received data known to be fraudulent, and it's very hard to perform a true validation because ground truth often isn't available. But on these data, on the fraudulent data, we attempted to perform some kind of validation on our tools. We also ran a few workshops to optimize the shape and form of it and see how it works. And over this process, we also collated this into a paper, as you can see on our right, that's published in Research Synthesis Methods. So anyone interested in our process of developing the tool can look there. Thank you, Kylie. Next slide. And we have one paper published on the process of developing the tool, and we have a companion paper that contains the step-by-step instructions, also in Research Synthesis Methods. That is really the meat of the study and how to use the IPD integrity tool. And so what is the IPD integrity tool composed of? There are 3 main components, as you can see in the top half of this figure. And that is an instructions and decision guide that was published in the paper I just on the previous slide, a rating sheet to collate whether each item had no issues, minor issues, major issues, and an automation script, which is an R markdown, which you put your data into, and then you knit it, and it produces output that you can then use to evaluate a trial's integrity and score on the rating sheet. And so what kind of things are we scoring? So there are two kind of main domains that we look at in the integrity tool. And the first is aggregate data level checks. So this is often on published information that's out in the public domain. And this is stuff that already exists in previous integrity tools, but we deemed it to be really important. So it's included in this tool too. And that's things like looking at retractions, looking at expressions of concerns, whether the trialist was willing to provide IPD, whether they opened a line of communication, whether they had ethics, whether they're registered, whether the randomization described sounded appropriate, and plausibility. Are they publishing 10 studies every day and recruiting 1,000 people in a short time frame with no grant money? But then moving on to the right, the new component, that the novel element of our tool is individual participant data, so raw data checks of integrity. And we have 8 kind of main areas in which we do this. I'm just going to list them here, and then I'll focus on a couple because it's probably too time consuming to go through them in detail. But unusual or repeated data patterns, looking at baseline characteristics, looking at expected correlations, whether there are date violations, looking at the patterns of allocation in terms of randomization, and then internal consistencies. So has a patient who died also got follow-up data? Are there things that are logically impossible in the data? Looking at external consistencies, so do the counts in your data set match published counts? And looking at the plausibility of the data, so the ranges in the raw data or the rates of missingness, or a whole myriad of things. So yeah, I'm going to now just focus on a few of the, I guess, juiciest items, because they provide really nice examples of less trustworthy data where we've had serious red flags kind of uncovered in our data integrity tool. And this is focusing on the ICOMP meta-analysis that Kylie briefly mentioned a few slides ago. And that's looking at cord clamping interventions for babies. And in childbirth, as gestational age at birth increases, as the baby becomes closer to term, it grows in the womb, and on average, on aggregate, you expect it to be larger and to weigh more. And on our left, we can see a graph as gestational age increases. On average, birth weight is also increasing. This is a typical trial. It's a really kind of reliable, consistent correlation that you'd expect to see in any trial with enough data. And then on the right, we had another trial, which I won't name, but relatively large trial, and there is absolutely no correlation that we could detect between gestational age at birth increasing and the child's birth weight also increasing, which based on clinical consultation is highly implausible. So this trial had a red flag for this item. Interestingly, there were also many clashes with external consistency. The counts we had in our data didn't match their published counts. When we queried these discrepancies, the trialist simply replied with an amended data set and no further explanation, which we found inherently incredibly untrustworthy. And this trial ended up being excluded from our meta-analysis after multiple kind of red flags emerged. And another item we look at is the integrity of randomization, so the patterns of allocation. Here we can see a plot, and on the x-axis is time. So as you go further right, time is, the date is getting later and later. And on the y-axis, we can see the cumulative count. So as you would expect, as the trial progresses, more and more people are getting randomized to either treatment. And when the pattern of allocation in a trial is 1-to-1, you would expect, especially in a large enough trial, these two lines, the blue line for treatment and red line for control, to interweave. But as you can see here, at just before halfway, the treatment starts getting more and more randomized to or more and more people are allocated into the treatment rather than the control, which is flagging some issue with the integrity of randomization potentially in this trial. And then I think a final integrity domain, or not a final one, but another one that I'm just going to talk about or focus on in this tool is randomization by the day of the week, which is a thing we look at when it's a non-pressing intervention. You would often expect less people to be randomized on Saturday and Sunday, and you would hope that equal amounts of people are randomized into either group on each day. This would be a potential red flag. It would be something you'd potentially query with the trialist here, because we have roughly equal amounts of people randomized on a Sunday as a Monday and Tuesday. It's possible this is innocent. It was a non-pressing intervention, but that would be something that would be flagged in our tool. And so moving on, we have a myriad of, we have quite a few items that then you can rate. And to really simplify the process of evaluating the integrity of a trial, there's a kind of decision tree, choose your own adventure, that is published in our paper. And it also can triage the process a bit, so you don't have to do every integrity check if you follow a certain route. So we start with, is there a retraction notice for this trial? If yes, the trial probably shouldn't be contributing to the evidence base and you can stop your integrity checks. If no, were there any issues identified across the items on the data integrity tool? If no, study can be included. If yes, then you need to evaluate whether there are major issues, minor issues, and whether they can be resolved after contacting the trialist or considering reasonable explanations. If no, many major issues remain, then the trial probably is not trustworthy and it shouldn't contribute to the evidence base. If no, some minor issues remain, then one needs to make a circumstantial decision based on the violations and whether they believe they can trust the trial. Yeah, I think that's our tool. If you could go to the next slide, Kylie, thank you. And then, yeah, so harking back to ICOMP, one of the IPDs that we focused on for piloting our tool, we had 61 trials provide IPD. So it's a pretty large IPD meta-analysis. And of these 61 trials, 55 had at least one integrity issue identified, but kind of to be expected. They were mostly minor inconsistencies and they could be resolved via consultation with the trialist. However, of the 61, 3 IPD trials were excluded due to major integrity concerns. And so in these 3 trials, we had multiple major discrepancies between the IPD and published data, often for primary variables like death. So we had different death counts to what was published. And there were many other red flags, such as the one I covered before, including a lack of association between variables that are known to be highly correlated. And yeah, so those 3 trials were removed and hopefully it improved our evidence base. And then when do we use the tool? So as Kylie mentioned, the tool was developed with the motivation of using it for individual participant data meta-analyses. But since publishing and giving a few talks on this tool, we've had a lot of interest from a lot of different stakeholders in a lot of different areas. And because it's a tool that simply evaluates the IPD of a trial, we realized that there was interest in also applying this across many other domains. So this could be in aggregate meta-analysis to scrutinize a particularly questionable trial, for journal submissions, when trustworthiness is a concern generally about a published study, and potentially for central monitoring of multi-center trials as well. And so now we have a little slide about tool use, a little question about tool use. And we were kind of interested in when you would like to use your integrity tool. These talks are nice because we always get a sense of the temperature for what people are most interested in and can help us optimize the tool in the future, hopefully.
Select multiple for this one, Sol, or it's just one?
Oh, good question. I don't know. I hope you
Seems
can.
like it's just one.
Okay, that's all right. So pick
Yeah.
your most important one. Okay, multi-center randomized trial. That's cool.
Go handle.
Interesting. All right, so maybe I'll click on, I think, no one selected other. So
Yeah, we have a follow-up question, but I guess it's not relevant with 0%.
But I guess people can think about it and let us know.
Yeah. Yeah, maybe we can go into that in the discussion period. Okay, so future directions. I think it's important to note that the research field and probably in turn fraudsters are not a static entity, and we too are hoping to be static and to optimize our tool and to iteratively improve it and to adapt with changing technology and cultural trends and whatnot. And yeah, the intention of this tool is to be this circular iterative thing that's constantly updating and being optimized, particularly with things like artificial intelligence and the act of simulating data becoming more and more accessible. We are hoping that our tool will be refined to start catching those kind of things as they have larger uptake and are more widely used. We're also kind of interested in making sure the tool is known about and applied widely. And it's nice that the Cochrane review team have endorsed the IPD tool in their policy for managing potentially problematic studies. And we've also had interest from a bunch of stakeholders. So Cochrane, as I mentioned, who's expressed interest, multiple publishers have contacted Kylie, hoping that they might be able to use the tool in certain instances for evaluating manuscripts that have been submitted for journal editors. Other researchers have expressed interest, and we've presented at research organizations like AMOS. And Kylie was actually nominated as a finalist in the Einstein Award recently, which was a nice little bonus to spread our visibility. A hurdle in making our tool more widely accessible is we presently use R Markdown to knit a report. And a problem with this is it requires some R skills to format and wrangle the data. And it can also take 20 to 40 minutes of coding and formatting to get output for the first trial in individual participant data meta-analysis. The first trial is the longest trial. Every trial after is proceedingly more quick, but that is a hurdle for adopting the tool. And what we're hoping to do about it, so we've actually kind of accessed the skills of Jim Cook, who works at the University of Sydney. I think I've forgotten what his team's called, but they're data
digital innovation or something.
Digital innovation, yeah, nice little catchphrase. And he has been really excited about the project too, and is a bit of a tech wizard. And using his team skills, we're hoping to make the tool more accessible so we don't need specific coding. And that's through developing a point and click interface on the application and website. And we're hoping that this in turn will also reduce the average coding time per trial. And a thing that we've also begun investigating is using AI to accelerate the variable selection and data wrangling process of integrity, so it can be more and more automated. And so what's the impact of our tool? We're hoping that it will filter out untrustworthy studies from the evidence base, and this can lead to more reliable, trustworthy evidence to inform guidelines, policy, and practice. And in turn, there's less research waste. It can hopefully protect patients from situations like the ivermectin example we saw earlier. And in a climate of increasingly skeptical of science, hopefully systems like this will foster public trust in research. Okay, I might hand back to you, Kylie.
Yeah, so before we go to discussion, I might just briefly add that, you know, the IPD integrity tool is intended as a sort of a filter. You know, ideally we would want to prevent the pollution in the first place. So we need systemic upstream changes to prevent this pollution. And just very briefly, we've thought about ways, you know, education is an important one, and awareness of the issues. I guess we need to address the publish or perish culture, which encourages this sort of behaviors, emphasizing quality over quantity. There is a board game called Publish or Perish as well, if anyone's interested. But I guess essentially, in the words of Doug Altman, we need less research, better research, and research done for the right reasons. So also offering credit and protection to whistleblowers if they have justified integrity concerns. We don't want people going on a witch hunt, you know, but at the moment, you know, you're not very popular if you're a whistleblower. It's not the best way to build a career and make friends. So, and so we want to also foster a culture that values self-correction. So, you know, if someone notices an error in their work, that, you know, you don't really want to get your study retracted because of the negative connotations, you know, can threaten their career, their reputation. But actually, you know, there are a lot of, I think I was going to say, there's an estimate that 20 to 60% of retractions are due to honest errors by authors. So humans are fallible. We should encourage self-correction and reduce stigma around retractions and use them as learning opportunities. And we've briefly mentioned journals and publishers. They can use these screening tools. They should promote open science, so data sharing, which enables us to actually do these checks. And as evidence consumers, it is important to have a healthy skepticism. Don't just believe everything you read. And we encourage the use of an integrity screening tool. So the world is a dangerous place to live, not because of the people who are evil, but because of the people who don't do anything about it. So we encourage people to do something about it. Just want to acknowledge this is our team, some of whom are on the Zoom today, I think. So thank you for the contributions. And if you are interested in trialing the tool, we'd love to hear your feedback. As I said, it's an iterative tool which we are refining as we go. So please do email us. And now I think we've just got some questions that may stimulate some discussion here. But do we have? Yeah, over to you guys now to see if you have any specific things you want to talk about. Thanks.
Thanks so much, Kylie and Sol. Very thought-provoking, and I think most of us here on this call are clinical trial statisticians with access to the individual patient data for trials we're working on. So I think it would be you know, really interesting to be trialing your tool on our own data sets, which of course we never have any sorts of serious errors in them, but it would be interesting to do that. So while waiting, please put up your hand or type a question in the Q&A, and while we're doing that, I might ask, I'm sure this is in your paper, but what was the process of validating your tool? For example, you know, getting a selection of known fraudulent, to use a better word, or fabricated studies and those not, and blindedly assessing them, or however you might have done it?
Yeah, so we have been contacted by some journal editors who asked us to check data sets. So those editors were able to share with us, one, that they had known ended up being fraudulent or retracted. And we tried to match them with studies which we assumed were truthful. And so assessments were blinded as well. So that was one part of the validation. Also played around with generating some data sets using AI, which was quite fun, just to test the tool there. So it was sort of a preliminary validation, something we want to do more of. But again, it's always, how do you know the ground truth? So how do you know if a study's made up or not? And people are getting better at making things up, I think. So that's a challenge that we have. But yeah, if you have any suggestions of other ways to do validation, that'd be great.
Sure. Okay, thanks. We've got a question in chat. Also, Christy's KR, I assume that's you, Christy. So please fire away. Your neighbour.
Hi, Christy.
Hi, guys. I just had a question about whether the tool has resulted in anything that's happened on retractionwatch.com, or what's your biggest implication of your tool being used other than excluding a few trials from an IPD?
It has not been. I have contacted Retraction Watch. I need to follow up on that. But at the moment, it's still sort of preliminary. So we are trying to promote it a lot, doing webinars like this, but also sharing with other people that are doing IPD meta-analyses. Also those like journal editors and publishers. Solid mentioned, we've contacted quite a few of them, but at the moment it's still sort of relatively in its infancy. But yeah, hopefully we can get it out there more broadly.
Okay, we have a question from Liliana in the chat. Can you see a future where IPD are required to be provided with publications, I guess with the submissions of RCTs?
We had a meeting with the publisher of Karga, who was very interested in setting up a system like this, where IPD was provided and they systematically evaluated all data that was submitted to their journal. But one of the hurdles is the scalability of having a tool that's so generalizable that it can just be replicated en masse to everything that's submitted.
But I think just to add there is definitely a move, as we know, towards data sharing, open access, transparency. So there are increasing regulations. You know, some journals already do require IPD to be shared. So it's just a bit tricky in terms of keeping those data confidential and making sure that they're not misused, I think. But that would make life a lot easier if it was just automatically provided.
Please fire in with more questions, but I've got a few, so I'll just fire away. I'll keep firing away until someone else comes in. Like students now use, when they submit assignments online, they use the Turnitin sort of plagiarism detection tool, for better word. So they do that prior to submission, and then they can modify their assignment and then submit something that passes the plagiarism test by that tool. And the university doesn't see the initial submissions; they only see the final one that they've submitted. Do you see that's a potential here too, that your tool becomes available and then a pathological data fabricator will go and check their data using your tool and find where the tool picks up and then change that bit so it passes? You'd have to be very committed.
It is a challenge. I mean, I sort of always think of it like doping in sport. You know, it's always going to happen. People are going to be one step ahead, but you've got to try something, and you do have to be really committed, because I guess that's when also like the other things come in, like ethics approvals and registration, and you've still got to have all that. Yeah, but it is an issue. Did you want to add anything, Sol?
Yeah, I think too, because there are certain things that the person maybe checking their data against the integrity tool wouldn't be sure of, such as what expected correlations are going to be present. And it is possible that you could simulate data really effectively and have a really plausible data set in the end. But I guess the hope is that knowing that it will be probed will instill some level of fear that would dissuade it, or at least make it so difficult that only a much smaller minority will be game to risk rolling the dice and making explicitly fraudulent data. Because you not only would need really good skills in simulating data, but you also need domain knowledge, and you need a kind of whole array of things that would be able to defend yourself against being caught, I think, that are hard to anticipate.
I guess you need sites as well. So you've got to say where you recruited. And if those sites read the paper and they're like, hang on, we don't know about this study. So yeah, I guess it's looking at all the different angles.
Yeah, I'm sure it will not stop people, but at least dissuade them and make it seem like a risk.
We had a talk in our Victorian Centre Biostatistics by Ben Mole from Monash University about sort of the data integrity crisis. I think the area was maternal and reproductive health or something or other like that. I don't remember exactly.
Yeah.
I saw he's one of your collaborators there, but he was talking about the whistleblower effect, that journals, they wouldn't take him seriously. They wanted to dismiss it. Have you had experience with journals and attitudes to, or potentially how you'd think about it for attitudes to the use of your tool to be informing journals that there could be potential problem with papers that they've published, and how you might negotiate that delicate conversation?
It's really very delicate, and we've thought about it a lot because we don't want to be the police. But our main goal is we want to trust the data that we rely on. So we are not looking at intent. We don't accuse anyone of fraud. We just sort of are looking at, do we trust these data enough that we would include them in our analysis? And I think we tread quite carefully. Ben, I guess Ben is sort of further along in his career and doesn't find as much. But yeah, it's a delicate balance, especially when we're building collaborations and us saying to people, hey, can you share your data with us? But we're going to scrutinize them and then call you out if there's something wrong. So we also are really careful about, we don't name and shame and publicly put this information in our, we will disclose it upon request, because obviously we want to be transparent. We don't want the data to be reused. And we did actually have a correspondence in the Lancet about this, where Ben Mole wrote a letter about it, and we responded because he thought we should publish our integrity results, and we said we prefer a more nuanced approach, balancing transparency and delicacy of the situation. So definitely something we've thought about a lot.
Yeah, he's definitely, from what he was saying, he's going with all guns blazing. Ben, I'm not sure if you're on the call here today. He may be here. I'm not sure. Lyle Gurran, who he's worked with. Yeah. Now, I just got a message that some people are trying to use some Teams features that they're not able to. So apologies, Kylie and Sol. I'm suspecting some people are trying to ask questions, but the features are not enabled for there. So that could be why it's not many people there. In terms of, how familiar with the John Carlisle situation in anesthesia? Because I was there at an anesthesia meeting where the results got presented, and then on the day that his name and shame list went up, and I was sitting next to someone who I won't name, who was like number 15 of the worst offenders. But it turns out that there are a lot of minor issues were flagged there. So his tool had a high false positive rate. But he was advocating at that stage that the journal use his tool for every submission. So his tool was just looking at the his tool was looking at the baseline comparison of sort of treatment A versus treatment B on a whole lot of variables. And so it was producing some false positive things. So I don't know how much that's being used today, or how do the other tools compare to yours? Is yours the only individual participant data tool and everything else is aggregate? Is that correct?
In terms of a standardized systematic tool, yeah. So there's things, John Carlisle has his sort of specific checks, but in terms of a tool, yeah. The only one. There is another one in development by Cochrane as well, but that'll be another couple of years before that's
by Cochrane.
Yeah,
Cochrane collaboration.
Yeah.
Oh, right.
Yeah.
Okay. Yeah.
So, Sol, did you want to comment on the Carlisle?
Yeah, I don't think it's necessary. I guess that sounds like his criterion was pretty low for bad integrity. At least when we've used the tool, I think our criteria for finding a data set not trustworthy was pretty high. Like, we were incredibly distrustful of the studies we excluded. They had they had so many red flags coming up. Everywhere you looked, almost there was a problem at the aggregate level, at the IPD level. Whether they were intentionally fraudulent or whether they just had incredibly bad practices, it's not clear, but at minimum, there are enough inconsistencies and errors on top of integrity flags that we really didn't trust the things being excluded. I think that there is a murky in-between level where there are some errors and inconsistencies, and that's, I guess, a little bit subjective still, even though we have a guide. But I think in our experience, when we've included trials, there's been a whole range of problems with those trials.
I guess I'll just add, if for the ones that are in the grey areas, we do sensitivity analyses to see if it makes a difference, whether they're included or not. But yeah, it's people sort of want a tool that just says yes or no, and unfortunately it's really not that straightforward. It's much more complex than that, and like Sol said, it can just be really poor study design or methods rather than anything bad intent. So it's a tricky one.
Could you comment a little bit more on the studies with multiple red flags? You mentioned it a bit in your talk, but you elaborate a bit more on what you actually did about that. So you excluded them, but in terms of communication with the authors?
We always give them an opportunity. So the first step is contact them and say, look, we noticed that this doesn't match this. Just being objective, not accusing them of anything. And often you just won't get a response. But like Sol said, in that one instance, we did get a response where I think there was 20 inconsistencies between the IPD and the published data, and they just changed the data set but didn't say why it was wrong. So, and yeah, like a lot of the time, it might be that the inconsistencies might be because they used a different sample, so it can be explained as a small error. But yeah, we always like to give the trials the opportunity to explain what's going on.
Yeah, errors were in almost every trial from my experience of going through them. And it's understandable when you are a trial statistician doing 50 trials that, there's a rounding error somewhere or some minor inconsistency that's non-consequential. But these often, as Kylie said, when we contacted them, they went and clarified, and it was not a consequential discrepancy.
Right. So I'm not seeing any further questions. Last opportunity, everyone. Okay, then I think we might wrap up then. So please do get in touch with Kylie or Sol if you want to try out the tool. I will definitely be in touch. Very, very keen to give it a go on a couple of past trials. And thank you very much for speaking about a really important sort of hot topic issue facing the clinical trials statistician and the clinical research community generally. So thank you so much again, Kylie and Sol.